Yeah, so as you already know, I'm using human stem cell models to study Alzheimer's disease. And specifically, what we are interested in the protein tau, which is one of the main protein pathological protein aggregates in Alzheimer's brain. And what we are trying to do is we try to remove the tau protein expression from these stem cells. These are stem cells from healthy individuals. So we remove the tau protein, then we try to differentiate these stem cells into neurons in the dish, and then we try to study what's the effects of removing tau protein. At the same time, we also try to introduce a toxic insult, named basically just introducing amyloid beta oligomers into the cell culture that those cells might or might not have the tau expression, and then we trying to compare what's going on. So like whether the removal of tau protein is good, is bad for the protein for the neurons, and this has a very direct implication to what we are studying in Alzheimer's disease, because we know that in mice that if you remove tau protein, then those toxic amyloid beta would not be able to elicit those toxic responses in various aspects. But we don't know what's happening in human stem cell models, so that's what we are trying to see. So the first question we ask is, what happens if you remove the tau protein, right, without any treatment? So just want to see at baseline what happened to those cells. So we focus on several experiments, mostly imaging-based experiments, and we try to see, for example, the number of synapses among these neurons. We also try to see axonal transport along the axons of these neurons using a microfluidic chamber. We also try to look at neural outgrowth to see how fast, how slow, how complex the neurites are forming between the tau knockout and tau wild type cells, and and also functional aspects like electrophysiology. And so far we have seen that in the tau knockout neurons, you would have a decrease in the speed of axonal outgrowth. At the same time, we also see some functional deficits in terms of electrophysiology, that they are firing less action potentials. They are firing action potentials with a lower strength. So like the amplitude is smaller, but we saw the same number of synapse expression across these tau knockout and tau wild type cells. And then subsequently, in terms of the treatment with amyloid beta, so far we have seen when you first treat the neurons with toxic insults, you want to look at cell death first. But cell death is quite difficult to try to induce in the cell culture, especially in these iPSC-derived neurons. So we needed to have a pretty high concentration of amyloid beta to treat the neurons. And what we can see is that in the wild type neurons, of course, the amyloid beta will kill the cells, about 20 to 30% of the cells after 5 days. But with the tau knockout cells, we can see that there's a protection. So the neurons probably decrease by about 10% over 5 days at a pretty high concentration of amyloid beta. In terms of functional rescue, that's what we could see as well. But although we weren't using amyloid beta oligomers, we were trying to use brain-derived, so Alzheimer's disease brain tissue-derived extracts, and then we were trying to mimic, the kind of AD environment in the dish, and that's what we tried to do in terms of the electrophysiology. And then we also saw that in the tau knockout neurons, they were not really affected by the brain extract treatment as compared to the wild type.